Hello, and welcome back to AP Biology, Big Idea 2, Free Energy. We're in Chapter 6, Section 7, all right, talking about extracellular components and connections between cells that help coordinate cellular activities. We're going to start with the cell walls of plants. The cell wall is an extracellular structure of plant cells that distinguishes them from animal cells. The wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. On the level of the whole plant, the strong walls of specialized cells hold the plant up against the force of gravity. Prokaryotes, fungi, and some protists also have cell walls, but we will postpone discussion of them until Unit 5. A young plant cell first secretes a relatively thin and flexible wall called the primary cell wall. In actively growing cells, the cellulose fibrils are oriented at right angles to the direction of cell expansion, possibly affecting the growth pattern. David Earhart and colleagues investigated the role of microtubules in orienting these fibrils. Their observations strongly supported the idea that microtubules in the cell cortex guide cellulose synthase as it synthesizes and deposits the fibrils. By orienting cellulose deposition, microtubules thus affect the growth pattern of the cells. Between primary walls of adjacent cells is the middle lamella, a thin layer rich in sticky polysaccharides called pectins. The middle lamella glues adjacent cells together. Pectin is used as a thickening agent in jams and jellies. When the cell matures and stops growing, it strengthens its wall. Some plant cells do this simply by secreting hardening substances into the primary wall. Other cells add a secondary cell wall between the plasma membrane and the primary wall. The secondary wall, often deposited in several laminated layers, has a strong and durable matrix that affords the cell protection and support. Wood, for example, consists mainly of secondary walls. Plant cell walls are commonly perforated by channels between adjacent cells called the plasmodesmata, which will be discussed shortly. Okay. All right, next we're going to talk about the extracellular matrix of animal cells. Although animal cells lack walls akin to those of plant cells, they do have an elaborate extracellular matrix. The main ingredients of the ECM, or the extracellular matrix, are glycoproteins secreted by the cells. Recall that glycoproteins are proteins with covalently bonded carbohydrates, usually short chains of sugars. The most abundant glycoprotein in the ECM of most animal cells is collagen, which forms strong fibers outside the cells. In fact, collagen accounts for about 40% of the total protein in the human body. The collagen fibers are embedded in a network woven from proteoglycans. A proteoglycan molecule consists of a small core protein with many carbohydrate chains covalently attached so that it may be up to 95% carbohydrate. Large proteoglycan complexes can form when hundreds of proteoglycans become non-covalently attached to a single long polysaccharide molecule. Some cells are attached to the ECM by still other ECM glycoproteins, such as fibronectin. Fibronectin and other ECM proteins bind to cell surface receptor proteins called integrins that are built into the plasma membrane. Integrins span the membrane and bind on their cytoplasmic side to associated proteins attached to microfilaments of the cytoskeleton. The name integrin is based on the word integrate, Integrins are in a position to transmit signals between the ECM and the cytoskeleton, and thus to integrate changes occurring outside and inside the cell. All right, so now that we've talked about that, let's go to the plasmodesmata in plant cells. It might seem that the non-living cell walls of plants would isolate cells from one another, but in fact, cell walls are perforated with channels called plasmodesmata, which is singular term plasmodesma, from the Greek desmos, which means to bind. Cytosol passes through the plasmodesmata and connects the chemical environment of adjacent cells. These connections unify most of the plant into one living continuum. The plasma membranes of adjacent cells line the channel of each plasmodesma and thus are continuous. Water and small solutes can pass freely from cell to cell, and recent experiments have shown that in some circumstances, certain proteins and RNA molecules can also do this. The macromolecules transported to neighboring cells seem to reach the plasmodesmata by moving along fibers of the cytoskeleton. All right, so now we're going to talk about tight junctions, which includes desmosomes and gap junctions in animal cells. In animals, there are three main types of intracellular junctions: tight junctions, desmosomes, and gap junctions, the latter of which are most like the plasmodesmata of plants. All three types of intercellular junctions are especially common in epithelial tissue, which lines the external and internal surfaces of the body. Okay. Let's talk about the cell. This is pretty much I would say the sum of all its parts, just a brief summary or overview of what we talked about so far. From our panoramic view of the cell's overall compartmental organization to our close-up inspection of each organelle's architecture, this tour of the cell has provided many opportunities to correlate structure with function. This would be a good time for you to go back and review some of the videos previously so that you can be refreshed. But even as we dissect the cell, remember that none of its components works alone. As an example of cellular integration, consider the microscope scene of what will be shown in your next class. You will be looking at some slides. All right? It helps to defend the mammalian body against infections by ingesting bacteria, the smaller cells, into phagocytic vesicles. The macrophage crawls along a surface and reaches out to the bacteria with thin pseudopodia called filopodia. Actin filaments interact with other elements of the cytoskeleton in these movements. After the macrophage engulfs the bacteria, they are destroyed by lysosomes. The elaborate endomembrane system produces the lysosomes. The digestive enzymes of the lysosomes and the proteins of the cytoskeleton are all made on ribosomes, and the synthesis of these proteins is programmed by genetic messages dispatched from the DNA in the nucleus. All these processes require energy, which mitochondria supply in the form of ATP. Cellular functions arise from cellular order. The cell is a living unit greater than the sum of its parts. And this is where we're going to stop for today. And the next time we meet, I believe we will be in chapter 7, talking about membrane structure and its function. Until next time, ta-ta.